So in this video, we're going to be looking at blood types, talking about the various different types and some of the biology behind them. And of course, most people are aware that within humans we have a handful of different blood types. This wasn't actually known until about 1900, and actually won this guy Carl his Nobel Prize. But what we're going to do is we're going to head in there. Now, before we get into the discussion, let's get some basic terminology down. What is an antigen? An antigen is something that basically can stimulate an immune response, and typically we're looking at the formation of antibodies made by white blood cells that will react to it. But antigens are very complex molecules on the surface of cell membranes. That's one group of antigens. And in this case, when we're talking about red blood cells, we're talking about a very specific group of antigens. Now, an antibody is a protein made by plasma cells. And plasma cells are basically B lymphocytes. We're going to learn more about that when we get to the immune system. That basically B lymphocytes, when they become activated to make antibodies, they change their name. They take on a new identity, and we call them plasma cells. And antigens stimulate the production of antibodies, and when antigens interact with antibodies, it's very much like a lock and a key, that they're specifically fit each other. Another term I want to introduce is agglutination. And agglutination is when I do have that binding of antibody to antigen, and typically we see this resulting in clumping. So if this is red blood cells that have been mixed with an antibody against their antigen, they'll clump together, and we'll call that agglutination. So let's start to have a look at the different blood types that we have. Most people are familiar with that we have type O, we have type B, we have type A blood, and we also have type AB. Now, what's the difference between them? Now, I got to point out that just to be completely upfront here, is that red blood cells, like any other cell in the body, have a lot of fuzzy bits sticking out. Cells always are fuzzy on the outside, almost like a fuzzy tennis ball, with things like glycoproteins, glycolipids sticking out from them, or if you're familiar with chia pets, they're kind of like chia pets. But we're really interested in one group of these. And the group that we're interested in is a particular backbone right here. And we can see in this basic diagram that if we have a look, let's look at type O blood here. You notice here's the backbone of type O. Here's the backbone of type B. Here's the backbone of type A. And with AB, you have molecules of both the A type and also the B type. But if you look carefully, you'll notice that the backbone in all of them is identical. The only difference is, do you have a side chain attached? And if so, what is the side chain? And that's what really distinguishes. You can see here in type O, you just have the basic backbone. There is no side chain. In type B, we have the same backbone, but here we have a different one. In this case, it happens to be a galactose. In type A, same backbone as we've seen. The only difference is that we have this N-acetyl group right here as the side chain. And if you have AB blood, it means you have antigens which are of the type B and the type A are both present within you. Now, not surprisingly, someone who is type A, we refer to them as type A, has the A antigens. Someone with type B has the B antigens. Someone with AB has both of them. And a type O has, now they say neither antigen, but you remember they have the basic backbone. And of course, the frequency of these different blood groups varies. Often when we're talking about blood types, we're talking about it within the context of basically blood typing. How is it done? Well, basically how it's done, and sadly, if we were able to do our hematology lab, we would have been able to try this, is you basically have two wells, you put in drops of blood from an individual. And then what you do is you have serums that are basically contain antibodies against the A antigen and antibodies against the type B. And you have the two wells of blood. So I'd have the two containers that are full of blood. I just put a couple drops of blood in each of the containers. Then what I do is, in one container, I add these antibodies. In this one, I add those antibodies. And as I said before, what happens is you have the antigens that are on the outside of the cell. And it's like a lock and a key. If the antibody happens to fit that antigen, then what happens is cells will start to clump together. So we'll get a clumping or an agglutination. And this will be held together by the antibodies, which will basically act like a Velcro and hold them together. Now, if that's the case, I'll start to see the clumps that are occurring. So here we can see that I've added the anti-A and the cells have clumped, which tells me that the antibodies in here have interacted with the cell membranes of these cells and they have basically Velcroed them together, and I get clumps. Here, when I tried the anti-B, so let's say this is the cell, and that's the shape of its antigen, and of course antibodies and antigens are like a lock and key. So let's say this antibody had a different shape. Well, that doesn't fit that, so therefore there would be no clumping together, and I don't see any clumping together happening here. So this tells me that this is type A. Same thing if it was type B, it would not react to the antibodies, and we don't see any clumping. Type B, there's clumping, and if I happen to have blood cells with both A and B, I would get clumping in both. Now type O, because these antibodies are against the specific antigen, remember the difference between the antigens is whether you have that backbone or not, and that's enough to be recognized as different. There's no clumping, so that's type O.